Hello, this is Frank from Intel Minds, and today we're going to be talking about 2.8 cell respiration from the IBDP program. The main idea is that cell respiration provides energy for the functions of life. Cell respiration is known as the controlled release of energy from organic compounds to produce ATP. There are two types of cell respiration, which are aerobic, involving oxygen, and anaerobic, which do not involve oxygen. The formula is shown below as C6H12O6, glucose, plus 6 molecules of oxygen gas will form 6 molecules of carbon dioxide and 6 molecules of water, and also ATP energy. ATP production. So ATP, or adenosine triphosphate, is composed of an adenine nucleobase, a ribose sugar, and 3 covalently bonded phosphate groups. ATP can be hydrolyzed to form ADP, which is adenosine diphosphate, plus inorganic phosphate, which is known as PI. When these covalent bonds are broken between the second and third phosphate group, it releases large amounts of energy that can be used for the cell to complete metabolic processes. So ATP becomes ADP plus PI, is known as dephosphorylation, and releases energy. ATP is immediately available as a power source for cell processes as this is broken down, but cell respiration also involves the use of energy from organic compounds to synthesize ATP using ADP and PI, and once these are joined together, they will also require energy. However, breaking them back down will release high amounts of energy for the cell to be used again. Anaerobic respiration. So anaerobic respiration occurs when oxygen is absent, and this pathway begins with glycolysis, which is the breakdown of glucose in the cytosol. Glucose, which is a 6-carbon compound, will break down into 2 pyruvate molecules, which are each a 3-carbon compound. It also forms NADH as it is reduced from NAD+, as it is a hydrogen carrier, and adding a hydrogen will basically be a reduction. It generates a net yield of 2 ATP molecules. So, in animals, pyruvate is converted into lactate or lactic acid, and in plants and yeast, pyruvate is converted into ethanol plus carbon dioxide. When studying the lactate production in humans, we realize that as we exercise at a very high intensity, not enough oxygen is available for us. Therefore, we must resort to anaerobic respiration also. So the body begins to break down glucose anaerobically, which is a carbohydrate. These carbohydrates will undergo anaerobic respiration, unlike lipids and proteins, which usually do not. This produces lactate, which causes muscle fatigue. So the graph on the left shows that a change in cell respiration conditions will also affect the energy demand as it increases. So aerobic respiration involves the consumption of fats, and this is decreased at high levels of exercise intensities, and the breakdown and consumption of sugars is increased. Furthermore, we can interpret that lactate levels increase at higher levels of exercise intensity. Aerobic cell respiration mainly focuses on the mitochondria, and it requires oxygen to be present. It also starts off with glycolysis that we previously talked about as the preliminary stage. However, as it uses oxygen, it is able to form much more ATP molecules, around 36 net molecules from these reactions that involve the link reaction, the Krebs cycle, and the electron transport chain. Pyruvate is broken down into water and carbon dioxide during aerobic cell respiration. Finally, we have respirometry, which is a lab that involves the analysis of respiration rates in germinating seeds or invertebrates. So this uses a respirometer, which is a device that determines an organism's respiration rate by either measuring the rate of exchange of oxygen gas or the exchange of carbon dioxide. In this lab, we will use germinating seeds that are enclosed in a sealed test tube. We'll have an alkali substance, such as potassium hydroxide, that is used to absorb the acidic CO2 gas so that only changes in oxygen levels are observed. And this ensures that our independent variable is highly measured and highly controlled. So the middle apparatus shown here is a U-tube manometer, which is used to measure changes in the meniscus level as we can observe here, and basically interpret this as the change in oxygen gas levels. Finally, the factors that affect respiration rates during this lab would be temperature, hydration levels, light intensity if we are using plants and germinating seeds, or activity levels of the invertebrates if we are using them for this lab. Thank you for watching this video, hope you enjoyed it, and make sure to like and subscribe to our channel. Until Minds.